Hello friends, my name is Dr. Santosh Abraham and I'm working as a specialist registrar in Scarborough General Hospital. So this is an important topic in the SE exam, namely osteoporosis. So today I'll be discussing about the physiology of osteoporosis and also the pathology to some extent. So bone is a special form of connective tissue and it has got a collagen framework impregnated with calcium and phosphate salts. So they are called hydroxyapatites. So basically it is collagen, impregnated with calcium, and it's involved in overall calcium and phosphate homeostasis. So bone is a dynamic tissue and old bone is constantly being resorbed and new bone is formed, permitting remodeling. So the remodeling allows the bone to respond to stresses and strains. So bone is a living tissue and this is well vascularized. So during the fetal development, most bones are modeled in cartilage and then they are transformed into bone by ossification. So this is called end-chondral bone formation. But the exceptions to these are the clavicles, the mandibles, and certain bones of the skull, in which bone is directly formed from the mesenchymal cells. So this is called intramembranous bone formation. So during growth, there are specialized areas at the ends of bones which are called epiphyses, and the epiphyses are separated from the shaft of the bone by a plate of cartilage called the epiphyseal plate. This plate is constantly dividing and proliferating. So as this plate lays down new bone on the end of the shaft, the length of the bone increases, and width of the plate is proportional to the rate of the growth. It's very important. The width of the plate is actually proportional to the rate of the growth than the length of the plate. So the width is affected by a number of hormones, but most markedly by pituitary growth hormone and IGF-1. So width is affected by growth hormone and IGF-1, and width is important for the rate of growth. And linear bone growth can occur as long as the epiphyses are separated from the shaft of the bone. But the linear growth will stop once the epiphyses unite with the shaft, and this is called epiphyseal closure. So summarizing, there are two pathways of bone formation. One is end-chondral and the other is intramembranous. So intramembranous is basically for the skull, clavicles and mandibles. So the bone is directly formed from the mesenchymal cells, whereas in end-chondral you have a scaffolding of cartilage, and which is then transformed into bone by ossification. And then you have the epiphyses, which are the ends of the long bone, and they're separated by a plate of cartilage which is actively dividing, called the epiphyseal plate. And the width of the plate is proportional to the rate of growth. So the width of the epiphyseal plate is affected by growth hormone and IGF-1, and as long as the epiphyses are separated, linear growth occurs, but it will stop once the epiphyses unite with the shaft. So bone in children and adults are of two types: compact or cortical bone, that makes up the outer layer of most bones and accounts for 80% of the bone in the body. And then there is a trabecular or the spongy bone inside the cortical bone. It constitutes about 20%. So the compact bone, the surface to volume ratio is low, whereas in the trabecular bone the surface to volume ratio is high, and bone cells lie in lacunae in cortical bone. So here the bone cells in trabecular bone, the bone cells sit on the surface of the plates, and in compact bone it receives nutrients by way of canaliculi, but in cortical bone nutrients diffuse from the ECF, extracellular fluid, to the trabeculae. So in compact bone, nutrients are coming from the canaliculae. So nutrients are provided by Haversian canals, which contains the blood vessels. So around each Haversian canal, collagen is arranged in concentric layers forming cylinders called osteons or Haversian systems. So around the Haversian canals, collagen is arranged in layers and it forms cylinders called osteons or Haversian systems. So there are two types of bone: cortical bone, which makes up 80%, and that's on the outer layer of most bones, and trabecular bone or spongy bone, that's inside most of the bones, and that's 20%. So surface to volume ratio is low in cortical bone and high in spongy bone, and they receive nutrients by way of canaliculi and Haversian systems, Haversian canals in cortical bone, whereas nutrients diffuse from the extracellular fluid into the spongy bone. So the protein is made of about 90% is made of collagen, type 1 collagen. So it's also the major structural protein in tendons and skin. So what exactly is collagen? It is made up of triple helix of 3 polypeptides bound together, and they are very tightly bound together, and 2 of these are identical alpha 1 polypeptides encoded by a single gene, and the remaining is a alpha 2 polypeptide and it's encoded by a different gene. So 90% of the protein is type 1 collagen. So its structure is of a triple helix. So it contains 3 polypeptides bound tightly together, and it's got 2 of them are alpha 1 and another one is alpha 2. So the alpha 1 are encoded by the same gene, whereas the alpha 2 is encoded by a different gene. So bone formation, resorption. So the cells responsible for the bone formation are called the osteoblasts, and those responsible for bone resorption are called osteoclasts. What are osteoblasts? So they are modified fibroblasts. There is extensive growth factor regulation. That is, a lot of growth factors regulate their formation and their functions. So ossification specific transcription factors such as CBFA, RUNX2 also contribute to the differentiation of osteoblasts, and the RUNX2 is quite important in the osteoblast differentiation. So mice deficient for the RUNX2 gene develop skeletons without ossification, exclusively of the cartilage. So normal osteoblasts are able to lay down type 1 collagen and form new bone. So osteoblasts are responsible for bone formation, clasts are responsible for bone resorption. So osteoblasts are modified fibroblasts, and RUNX2 contributes to differentiation, and if you don't have the RUNX2 gene, the skeletons will be entirely made of cartilage. There will be no ossification, and osteoblasts usually lay down type 1 collagen and form new bone. But osteoclasts, they are the members of monocyte family. Stromal cells in the bone marrow, osteoblasts and T lymphocytes express what is called RANKL on the surface. So it's called the receptor activator for nuclear factor kappa beta ligand. So it is the short form. So they express this protein called the RANKL. So when the osteoclast when these cells, mainly these stromal cells or osteoblasts or T lymphocytes, they come in contact with appropriate monocytes expressing rank, the rank is the rank L receptor. There will be two signaling pathways initiated. One is the rank interaction between the cell pairs, and the other is a mononuclear phagocyte colony stimulating factor pathway. So MCSF is secreted by non-monocytic cells, so namely by the osteoblasts, T lymphocytes and bone marrow, and it binds to its corresponding receptor on the monocytes. So here in this case, if the osteoclast and the osteoblast are interacting, the osteoblast which contain the receptor activator, the rank L, will bind to the rank on the osteoclast, and then there is a rank interaction between these two cell pairs. Also, the MCSF is secreted by the osteoblast here, and it binds to the corresponding receptor on the osteoclast. So this combination of these two signaling events leads to differentiation of monocytes. So the monocytes differentiate into osteoclasts. The precursor cells also secrete osteoporotograin. So this controls the differentiation of monocytes by competing with the rank for binding of rank L. So osteoporotograin competes with basically with the rank L. So it competes with rank. So rank L. So the rank is affected, the binding is affected, so that it actually down regulates the differentiation of these osteoclasts. So osteoclasts erode and absorb previously formed bone. So attached to the bone via integrins in a membrane extension called the sealing zone, and this creates an isolated area between the bone and a portion of the osteoclast. So they attach the bone via integrins in a membrane extension called the sealing zone. So this sealing zone is an isolated area between the bone and the osteoclast. Now proton pumps then move from endosomes into the cell membrane, close to the isolated area, and they acidify this isolated area to approximately pH 4.0. So osteoclasts attach to the bones via integrins, and this area is called sealing zone, and the sealing zone is actually an isolated area between the bone and the portion of the osteoclast. So proton pumps actually they acidify this area to approximately pH 4.0, and this sealed off space formed by the osteoclast, it resembles a large lysosome. So the acidic pH will dissolve the hydroxyapatite, and acid proteases are secreted by the cell breakdown collagen, and it forms a shallow depression in the bone. Now these products are digested, and then they are endocytosed, and then they move across the osteoclast by transcytosis, and then finally they are released to the interstitial fluid. So the sealed off space, which resembles a large lysosome, inside that the acidic pH dissolves the hydroxyapatite, and also the acid proteases dissolve the collagen, and the products of digestion are then moved across the osteoclast to the interstitial fluid. The collagen products, breakdown products, have pyridinoline structures, and these pyridinolines can be measured in the urine as an index of the rate of bone resorption. So that's very important. So at the cellular level, there is some regulation of osteoclast formation by osteoblasts via rank and the MCSF OPG mechanism. So specific feedback mechanism of osteoclasts on osteoblasts are not well defined. The bone remodeling process is primarily under endocrine control. So PTH accelerates bone resorption, and estrogen slow bone resorption by inhibiting the production of cytokines. Intracerebroventricular but not intravenous leptin decreases bone formation. So this finding is consistent with observations that obesity protects against bone loss. And most obese humans are resistant to the effects of leptin on appetite. So thus there may be an neuroendocrine regulation of the bone mass via leptin. So the Wrangle-Rank and the MCSF OPG mechanism that is regulated at the cellular level. So the osteoblasts mainly regulate the osteoclast formation actually by these pathways. And leptin, the intracerebroventricular leptin, decreases bone formation. PTH accelerates bone resorption and estrogen slow bone resorption. So what is the role of estrogens in bone dynamics? So bone loss in women accelerates rapidly in the first years after menopause. So the lack of gonadal hormones is thought to upregulate osteoclast progenitor cells. So when there is estrogen deficiency, there is increased expression of Wrangle by osteoblasts and decreased release of OPG. So increased Wrangle results in recruitment of high numbers of pro-osteoclasts as well as their increased activity. And the lifespan of the osteoclast also increases. So estrogen deficiency causes increased expression of Wrangle and decreased release of OPG. So increased Wrangle results in higher number of pre-osteoclasts as well as increased activity and lifespan of these osteoclasts are increased. So estrogen deficiency not only accelerates bone loss in postmenopausal women, but it also plays a role in bone loss in men. So estrogen deficiency can lead to excessive bone resorption accompanied by inadequate bone formation. So all these cells, osteoblasts, osteocytes and osteoclasts, they all express estrogen receptors. In addition to the estrogen affects bone indirectly through cytokines and local growth effectors. So if the body is having enough estrogen, the estrogen is replete, it will enhance osteoclast apoptosis via transforming growth factor beta, TGF beta. And estrogen also deficiency also sensitizes bones to effects of PTH. So estrogen deficiency will cause bone resorption. So osteoblasts, osteocytes and osteoclasts have estrogen receptors. In the estrogen replete state, it will enhance osteoclast apoptosis through TGF beta. In estrogen deficiency state, it will sensitize bone to the effects of PTH. So when there is no estrogen, T cells promote osteoclast recruitment, differentiation and prolonged survival via IL-1, IL-6 and TNF alpha. So when there is no estrogen, T cells via IL-1, IL-6 and TNF promote osteoclast recruitment and their differentiation. So the estrogen specifically inhibits IL-6 secretion because IL-6 contributes to recruitment of osteoclasts, thus contributing to osteoporosis. So IL-1, it has been shown to be involved in production of osteoclasts. So it is increased in bone marrow nuclear cells in overactivized cells. So administering this IL-1 antagonist to these patients, to these animals, prevents the late stages of bone loss actually, induced by the loss of ovarian function. So when there is no estrogen, it will result in more IL-1, and IL-1 will cause more osteoclasts to be produced, and more bone will be resorbed. So the WNT family, it's a very important family in bone metabolism. The WNT family is a highly conserved group of proteins. So this was initially studied in cancer, but they were found to have a role in intercellular communication. So now WNT signaling cascade is recognized as a critical regulator of bone metabolism. So mainly it affects the mesenchymal stem cells. So these are the progenitor cells of mature bone forming osteoblasts. So the WNT is an important regulator of bone metabolism. It affects the mesenchymal stem cells called the MSCs. MSCs form the osteoblasts. These are the progenitor cells. So MSCs are pluripotent. They can differentiate into adipocytes, chondrocytes, neurons, and muscle cells, as well as in the osteoblasts. So certain WNT pathways promote differentiation of MSCs along the osteoblast lineage. So there are specific molecules involved in this WNT pathway. So it has helped us to understand more about bone metabolism, and also for the development of new therapeutic targets for metabolic bone diseases. So WNT signaling pathway has got activation has it will progress along 3 pathways. So the canonical pathway involving beta-catenin is the most important. So when a WNT protein binds to an extracellular co-receptor complex called the frizzled FR, and also with the LRP5, the low density lipoprotein receptor related protein, it activates, it recruits another protein called the disheveled to the intracellular segment of the combined frizzled and DVL. So here beta-catenin comes into play. So when the WNT binds to the extracellular co-receptor complex, so the co-receptor complex containing the frizzled and the LRP5, it leads to, so when the WNT basically when it binds the frizzled and the LRP5, it recruits disheveled, and then the beta-catenin comes into play. So beta-catenin normally exists in a phosphorylated state, so it's important in intracellular signaling. So activation of the WNT pathway leads to dephosphorylation and stabilization of the beta-catenin, and the concentration of the beta-catenin in the cells increase. When it reaches a certain level, the beta-catenin travels to the nucleus. Then it activates the transcription of WNT target genes. And WNT signaling inhibits the expression of transcription factors important in the differentiation of MSCs, such as PPAR gamma, and promotes survival of the osteoblast lineage cells. So when the beta-catenin what happens is that when the WNT pathway is activated, it leads to dephosphorylation and stabilization of the beta-catenin. Hence the beta-catenin concentration increases inside the cell. After a critical level, it travels the nucleus. Then it activates the transcription of WNT target genes. And it inhibits the expression of transcription factors important in the differentiation of MSCs, such as PPAR gamma, and it promotes the survival of osteoblast lineage cells. So bone abnormalities linked to WNT pathway. Several several single amino acid substitution in the LRP5, single amino acid substitution in the LRP5 receptor has been associated with high bone mass phenotypes. Also, the mutant LRP5 receptor had an impaired interaction with the WNT signal inhibitor. Other missense mutations for LRP5 have been implicated in high bone mass diseases like Van Buchem disease and osteoporosis. So, if you have a mutation in the LRP5 receptor, usually the missense mutation or a single amino acid substitution, it leads to high bone mass. But there are loss of function mutations of LRP5 also. It results in severe congenital osteoporosis. So WNT antagonists. So the two most well known are DKK1 and sclerostin. So DKK1 is secreted by MSCs and binds to LRP5 and LRP6, and it inhibits WNT signaling. So serum levels of DKK1 positively correlate with the extent of lytic bone lesions in patients with multiple myeloma. So if the serum levels of DKK1 increase, it means that there is more resorption because the WNT signaling is inhibited. So this anti-AKK1 monoclonal antibody accelerates bone formation and increases the bone mineral density. So the anti-DKK antibody is now under development as a bone anabolic agent. So this monoclonal antibody accelerates bone formation, anti-DKK1. It also increases the bone mineral density. Other factors implicated in the pathogenesis of osteoporosis could be polymorphisms in the vitamin D receptor, alterations in IGF-1, BMP, that is a bone morphogenic protein, PGE2, NO, leukotrienes, collagen abnormalities, and leptin-related adrenergic signaling. Sclerostin, a product of osteocytes, has also been found to antagonize the WNT signaling pathway by binding to LRP5 and LRP6. So sclerostin antagonizes the WNT signaling pathway by binding to LRP5 and LRP6. So it's produced by the osteocytes. So romosozumab is a monoclonal antibody that is targeted against the sclerostin, and it inhibits the sclerostin, and it increases bone formation and decreases bone resorption. This has been approved for the treatment of osteoporosis in postmenopausal women who are at high risk for fracture by the Endocrine Society. So romosozumab is a sclerostin inhibitor. It's a monoclonal antibody against sclerostin. So collagen is degraded by proteases. These are called collagenases. So these are matrix metalloproteases. It can initiate cleavage of collagen at neutral pH, and these are the central enzymes for the process of collagen degradation, matrix breakdown, and bond remodeling. So there are three types of collagenases: MMP1, MMP8, and MMP13. So they are called respectively 1, 2, and 3 collagenases. So osteoblasts express collagenase 1 and 3, so MMP1 and MMP13. Resting osteoblasts, they secrete limited amounts of collagenase, and changes in the synthesis of collagenase correlate with change in bone resorption. So these collagenases have a critical function in bone remodeling. So cleavage by collagenase or splitting of the collagen by collagenase is an early and obligatory event. But most of the degradation of collagen is done by the osteoclasts. They secrete protons, H+ and enzymes like cathepsin K. These solubilize the mineral part of the bone matrix, and also they degrade the matrix. So cleavage of collagen fragments are then excreted. The cleaved collagen fragments are then excreted in urine. There are sensitive assays which can detect the N terminal or the C terminal of the fragments. So osteocalcin is released mainly during bone remodeling. So osteocalcin reflects bone turnover rather than bone formation. And glu-13-OCN, it's a newly studied molecule. It could serve as an endocrine hormone. It prompts insulin secretion, and also it increases the insulin sensitivity in peripheral tissues. So CCAT and enhancer binding proteins like beta and gamma and PPAR gamma, they play essential role in the differentiation of cell towards adipocytes. The MSCs could develop into adipocytes. So RUNX2 plays a central role in the differentiation of cell towards osteoblasts. So if that RUNX2 is disrupted or it is made inactive, it results in a mass of cartilage, so it results in disorganized chondrocyte, and there is a complete lack of bone formation due to an arrest of osteoblast development. So if the RUNX2 is getting affected, the osteoblasts fail to develop, and then there is the bone formation is also impaired. So Osteorix SP7, it is another important transcription factor, and it is required for endochondral and intramembranous bone formation. So RUNX2 also regulates Osteorix SP7. So it is the next stage in osteoblast differentiation. So it acts cooperatively with a nuclear factor of activated cells called NFAT. So NFAT is a transcription factor. It regulates osteoblast formation as well as osteoclast formation. So this CCAT, which I've shown you before, CCAT enhancer binding gamma and delta, which they can interact with the RUNX2, and they activate transcription factors called the ATF4. So ATF4 is important in the osteoblastic function. So in short, RUNX2 regulates SP7, and it also and the SP7 acts cooperatively with the NFAT. So then the RUNX2, which binds with the ATF4, it plays a central role in osteoblastic function. So osteocytes, they probably sense bone deformation as well as fluid shifts. They provide signals for the adaptive remodeling of the bone size and shape. So how can they sense bone deformation? It could be due to small strains which produce fluid shear stress in the canaliculi between osteocytes. So this will result in intracellular signaling through the ion channels, and or it could also result in production of biologically active molecules. So where there is bone microdamage, it contains this apoptotic osteocytes. So this will provide signals for initiation of bone remodeling by osteoclasts and consequent removal of the damaged bone. So when there is a bone damage, the osteocytes become apoptotic, and then they give signals for bone remodeling. They give signals to the osteoclast for bone remodeling, and they remove the damaged bone. So most of the hormonal factors that stimulate the bone resorption act on cells of the osteoblastic lineage. So they stimulate the synthesis and perhaps the release of RANKL and CSF1. These are essential for osteoclastic genesis or formation of osteoclasts. Osteoblasts also produce additional factors like cytokines, PGs, local growth factors. They regulate bone resorption. So the hormone factors which stimulate bone resorption are produced by osteoblasts, mainly the RANKL and the CSF1. Also others like cytokines, PGs and local growth factors. So there is another transcription factor called SP1. It is necessary for this osteoclast precursor to develop. So osteoclast precursor cell is a multipotent cell. As I told before, it is of monocytic origin. So it could differentiate into monocyte macrophages or dendritic cells or pre-osteoclasts. So the pre-osteoclasts, they fuse to form highly differentiated multinucleated osteoclasts like a syncytium, and these reabsorb the bone. Osteoclasts also contain large number of mitochondria because and hence they have high they can function well in oxygenated and hypoxic environments, and they are susceptible to apoptosis also. So osteoclast formation, the principal stimulator is RANKL. So it's a member of the TNF protein family. So it was originally identified as product of activated T lymphocytes, but it's also a stimulator of mesenchymal stromal cells, osteoblasts, osteocytes and hypertrophic chondrocytes. So when osteoblasts produce more RANKL, they help in the formation of osteoclast. So the production of RANKL is stimulated by PTH, 125 hydroxy vitamin D, prostaglandins and also many cytokines. So all these increase the production of RANKL. So mice who have no RANKL, they are deficient RANKL, they do not form osteoclasts and they have osteoporosis. And if mice are injected with RANKL, it stimulates osteoclast formation. These mice are, if they are injected with RANKL, it stimulates osteoclast formation and bone resorption. So RANKL is a membrane protein. So it is cleaved from the cell membrane and it's released as a soluble factor. Now what about Osteoporotograin? So it's a native inhibitor of osteoclastogenesis. It's also a soluble receptor for the RANKL, and it binds with this ligand and prevents interaction of the RANKL with its cognate receptor RANK. So OPG actually it binds with the RANKL, so thus it prevents RANKL being bound to RANK. So the PTH 125, the PTH vitamin D and the prostaglandin E2, which stimulated the RANK production, as I've shown before, which stimulated the RANKL production, will inhibit the OPG production. So there is a reciprocal stimulation between RANKL stimulation and OPG inhibition. So mice who have deficiency in OPG have osteoporosis, but which overexpress OPG have increased bone mass. So the bone resorption is tightly regulated by the combined actions of RANKL and OPG. So some designated cells in the osteoid, they form the dendritic projections. So molecules such as E11, GP38, MT1, MMP, they have an important role in this dendritic or canaliculi formation. So phosphate regulating endopeptidases, FEX, MEPE and DMP1, they regulate. So FEX, MEPE and DMP1 regulate biomineralization, while as the FGF23 or the fibroblast growth factor 23, it regulates renal phosphate excretion. FGF23 is not only elevated not only in osteocytes but also in hypophosphatic animals. And sclerostin is a marker of mature osteocyte, and it's a negative regulator of bone formation. So the receptor for RANKL is RANK, as I've told you before. So osteoclasts express this RANK, and their expression is induced by MCSF. So RANKL to RANK, it activates a series of intracellular pathways which will activate the NF-kappa beta and MAP kinase as well as the NFAT and the activator protein AP1. The TNF receptor associated factors TRAFs, particularly TRAF6, are ubiquitous in E3 ligases, and it binds the RANK intracellularly, are involved in RANK responses. So the main receptor for RANKL is RANK. So osteoclasts and other precursor cells express RANK, and the expression of RANK is induced by MCSF. So when the RANKL binds to RANK, it activates the intracellular pathways that in turn activates NF-kappa beta, MAP, NFAT and AP1. So mice deficient in TRAF6, like those deficient in RANK, develop osteoporosis actually. So these mature osteoclasts express RANK, and treatment of these cells with RANKL inhibits apoptosis and stimulates resorptive activity. So RANKL inhibitors, they inhibit the apoptosis and stimulate the resorptive activity. So if you treat them with RANKL, not the RANKL inhibitor, it inhibits apoptosis and stimulates resorptive activity. But if there is a RANKL inhibitor, the reverse happens. So MCSF is essential for osteoclast formation. So if you don't have the MCSF, you have osteoporosis. So both MCSF and RANKL must be present to form mature osteoclasts. So what does MCSF do? It enhances the RANK protection in osteoclast precursors, and it inhibits apoptosis of osteoclast precursors. So it enhances the rank production and inhibits the apoptosis of osteoclasts and mature osteoclasts. So the receptor for the MCSF is called the CSF1 receptor, or CSF1. So CSF1R, when it binds with MCSF, it activates tyrosine kinase activity in the receptor. So it further initiates a series of intracellular downstream events. So MCSF is important for osteoclast formation. So if you have a deficiency of MCSF, you have osteoporosis. So both MCSF and RANKL must be present in for the formation of mature osteoclasts. So MCSF basically enhances rank production in osteoclast precursors, and it inhibits apoptosis of osteoclasts. And the receptor for MCSF is called CSF1. So CSF1 and MSF, when they combine, it activates tyrosine kinase activity in the receptor. So in the next part, I will discuss more about the pathophysiology. Thanks for watching this presentation.